Your second worksheet, which is chapter 3, is due Friday. Yeah, 3. All right. At midnight or 11:59. So I'm getting the sense that fewer people are as far ahead on this one as the last one. Yeah. I'm getting the sense that maybe some people haven't even really spent a lot of time on it yet. Some people have. So since by this point last week, most people were this close to finishing the worksheet, and this week I don't think that that's true. I will give most of Friday as sort of a work day. I haven't gotten more than 1 or 2 questions on this worksheet, and I anticipate that people are going to have more than 1 or 2 questions. So on Friday, I'll spend maybe 15 or 20 minutes going through chi-square again as a whole class, and then I'll give the rest of the time to work on this worksheet, or you can start working on the worksheet for next week, or if you have additional problems you want to go through, we can do that as well. So come Friday with things prepared. Our first exam in this class is a week from Friday, so that is September 27th.
Yes.
Not the, did I say the 25th? I think it's on the 20th. I think we'll do the 27th.
I was going to say the analytical one is the 25th, which is the Wednesday.
All right, so I'll do the 27th. Make it gentle. I think it just has the week.
Oh, okay.
Yeah, so we'll make it on Friday. So what we'll do is today we'll actually get into chapter 4. Friday will take as a work day, and then Monday and Wednesday we'll finish up chapter 4 so that we're prepared for Friday to do the first exam. In terms of exam structure, what you should expect is 20 to 25 multiple choice questions, and then 1 short answer question for each of the chapters. And those short answer questions will ask you to sort of explain or demonstrate your knowledge of sort of the biggest concept in that chapter. So for chapter 2, meiosis and mitosis, like simply the processes of meiosis and mitosis would be our big concept. In chapter 3, it would probably be some sort of inheritance problem. Chapter 4, I don't know yet because I haven't gone through the material yet. So I know what the main concepts are, but I don't know what the questions will be. So yeah, I can give you a little bit more detail once I actually finish writing the exam. Questions on that? All right. There's something else I want to say. For the next 3 to 4 labs, you will need somebody in your group to have a computer. So next week's lab and then the lab for week 6 are going to be simulation based, which the alternative is that I actually make you breed fruit flies, or we can pretend to breed them on a computer. So breeding fruit flies means that somebody comes in every 18 hours to see if they're alive or not. I think most people would prefer computer work. So I forgot to mention it in lab yesterday, but discuss amongst your groups. You can have more than 1 computer, that's fine, but you'll need at least 1. And then after fall break, when we come back, we're going to start the sort of wet lab stuff, which means that we'll be actually doing, I think, cool stuff with things. So prepare yourselves. Okay. We are going to get moving unless there are questions. Awesome. Okay, so we left off here on Monday. Those of you who had lab yesterday know that we actually went through sort of the next piece of the PowerPoint in lab. So I'm going to skip the chi-square portion in class right now because some people have had it and some people have not. If you have lab tomorrow, I will go through how to do a chi-square with you at the beginning of lab. And then Friday, when we're back in class, we will do sort of one big chi-square problem together so that we can make sure that we're all on the same page with that. So we're just going to skip ahead. And you don't have as many slides as I do, so. Okay. All right. So when we're talking talking about Mendel, we often sort of talk about it in this way that's like, oh, this is all history and we know all this stuff and we're just demonstrating basic concepts. And we don't usually look at it from our current perspective because we've moved into a different sort of age in genetics where now we look at whole genome sequences and protein expression and all these other really cool things. But I did want to point out that we do still use Mendel and his principles in a couple of very important ways, usually when we're dealing with diseases and disorders, right? So we often sort of go back to what genes a parent has when we're trying to determine if their offspring may also be affected. And the way that we usually do this in humans is through sort of what we would consider family trees. We also call them pedigrees, right? Although I think people usually associate pedigrees with like dogs and horses and things like that. But we all have pedigrees and we still use them to sort of figure out what diseases or disorders sort of maybe coming to us from our ancestors, right? Why would we use pedigrees in humans instead of crosses like we do in fruit flies or other sort of lab animals? So if you're a fruit fly and I notice that you have like a really cool phenotype, I will take you out of a vial and I'll put you in a vial with the opposite sex.
Force two humans to make a baby. This
Yeah, right. So it is unethical to force one person who has a cool phenotype to mate with somebody else who has a cool phenotype to see if their offspring will also have that cool phenotype, right? That's something that actually we have done in the past, but in 2019, it's not okay, right? So it becomes a little bit more challenging in some ways because we can't just look at two individuals and figure out like what their offspring would look like. We have to sort of go back in their family trees and see those traits and see sort of how they move from generation to generation. If anybody knows a way to get around this ethical piece, then maybe you could find out cool things about people, but I don't see it happening anytime soon. All right, so we are going to look at pedigrees in lab. So again, some of you have seen these symbols already. We will look at pedigrees sort of continually throughout the semester. So I just wanted to walk through what the different pieces of a pedigree mean. So we usually, in most of our pedigrees, will see circles and squares. Circles and squares are indicating whether an individual is male or female. If we don't know, or it's undetermined, it would be a diamond. Okay, so this may be a baby that was never born. It may be an ambiguous phenotype, right? So we don't know whether it's actually male or female. If a person, because we're usually tracing a disease or disorder through a pedigree, we designate people as affected or unaffected. An affected individual would be somebody that shows the phenotype of that disease. So if you're tracing like cystic fibrosis, somebody who's affected by cystic fibrosis would be shaded, and they show red here, but usually it'll just be sort of gray or black. These are actually fairly rare to see. Okay, these would be sort of special circumstances where we actually know that a person is an obligate carrier or asymptomatic. You can put those marks on a pedigree, but usually if I hand you a pedigree, it won't have those. Okay, so that's something that we'd be looking at figuring out. Most of the time, a lot of people in a pedigree are going to be deceased, so we don't use this designation as much. The other sort of important ones are going to be over here. Pedigrees are set up generation by generation, so the highest, or like the top line, would be the oldest generation. The next line is going to be like a subsequent generation. Is everybody sort of familiar with how to read a family tree or pedigree? So if we have lines between a male and a female, it indicates that they have reproduced or that they are in some sort of relationship. It doesn't have to be a marriage line. Can be, doesn't have to be. So then from that horizontal line that connects two individuals, any vertical lines will lead to children. or offspring that they've had, right? So these two individuals had 3 offspring. If one of these individuals marries or has an offspring of their own, we would draw another line and then we get the next generation. These are twins, so if they come down from a single line and then split off, those would be twins. The one that actually we like to ignore, but is really important from a genetic standpoint, is consanguineous matings, or relationships that produce offspring that are closer familially than we would anticipate, right? Very common in dog breeding, hopefully not terribly common in human breeding. So this would be what we would call inbreeding, right? So what is the relationship between these two people?
Cousins?
They're first cousins, right? So parents, brother, sister, they outbred, good for them. They then each had at least one child, so they would be cousins that then apparently reproduced. So I'm from Maryland, right? And so we always poke fun at West Virginia for being inbred, which is super funny because Maryland allows first cousin marriages and West Virginia does not. And so then I moved to Illinois and then up here, and so I was asking somebody like, what's your West Virginia? Like, who do you make fun of for being like hillbilly? And somebody said, nobody, Dr. Brandt, we're nice up here. That's right. Cool. All right. So I don't know if that's true or not, but we did have some interesting families in the area I grew up in. Okay. So yeah, there were some like trailers with some family trees that looked like triangles and yeah. All right. I grew up in a very rural area. Okay. Let's see. What was I saying? Okay, so let's just. One of the really useful things that we see in a pedigree is what type of inheritance pattern we see. So there are times where diseases or disorders or inheritance of a particular trait is dominant. Sometimes it's caused by a recessive gene, and most of the time if we're talking about a disease, it's going to be a recessive trait, so we don't see it as often as if it were dominant, but we will see some examples of dominant traits that we want to trace through lineage. So here is an example. Okay, so here we have 4 generations. In this particular pedigree, we have 2 individuals who are affected by a condition. There are a few important things that we want to take note of. So when we are looking at an affected individual, one of the first pieces of information we need to figure out is how or are their parents affected and what would the genotype of the parent be? Okay. So we have an affected individual here. Are the parents of this individual affected? No. What about the siblings? Okay, so if we have 2 individuals who are unaffected and they produce an affected offspring, what type of disease or disorder is this? Is it caused by a dominant allele or is it caused by a recessive allele? Recessive, right? So these individuals, like it says there, are recessive, which means that they're carriers, they're heterozygotes. Right? And so this particular individual ended up inheriting both of those recessive alleles and is therefore affected. The other thing that we want to pay attention to is how often do we see this particular condition cropping up? Right? So here it's sort of every other generation. That is very characteristic of an autosomal recessive disorder, right? Because we assume we have unaffected individuals that are carriers, they produce an affected individual. Even if this affected individual makes it to reproductive age and has offspring, they are likely to reproduce with an unaffected individual, which is going to mean that most of their kids are unaffected. So it skips this generation, but they'll all be carriers, so that here we see it come back in the next generation. Okay, so that sort of every other generation thing is very characteristic of recessive traits. And the final piece here is that an equal number of males and females are affected, which tells us that it's not sex-linked. So it's on an autosomal chromosome. Okay, so 3 pieces. Are the parents affected? Is it found in every generation? And what is the ratio of affected males to females? And so those pieces should help tell you if it's autosomal or sex-linked, and we'll talk about sex-linked in the next chapter, and whether it's dominant or recessive. Okay? In any situation for an autosomal recessive disorder, disease, or trait, if you have 2 affected individuals reproducing, every single one of their offspring will be affected. Right? You could do the Punnett square and sort of figure that out. So here's what a dominant pattern looks like. So this would be a dominant condition or trait. And I keep saying disease or disorder. This could just be a trait. So as everybody's, maybe you haven't, has everybody heard of polydactyly, having extra toes or fingers? That is actually a dominant trait. So it would potentially show this type of patterning if we looked at it in a pedigree. So if your grandmother had 6 fingers, it's likely that some of her kids would have 6 fingers. That's not a trait that's going to make you unfit or make you die at a young age. So those individuals who are affected would likely reproduce and keep passing that trait on. So we see it in every generation. If we have an affected parent, we are likely to see affected offspring. We should never see an affected offspring who does not also have an affected parent. Does that make sense from a dominant standpoint? One of those parents has to have the dominant allele, which means they have to be affected in order for the dominant allele to show up in the next generation. That or somebody is lying about who their parents are. Pedigrees get weird, I'm telling you. Humans are not as innocent as we make ourselves seem when we're talking about families and reproduction. Questions here? Sorry, I'm also teaching a class about gender and gonads and sex this semester, so it's floating around in my brain. All right. Any questions on pedigrees? Okay. So again, you'll see examples. I think there's one example in the problem set this week of a pedigree, and I'm asking, I'm pretty sure, what type of inheritance pattern you see. Right, so again, oh, I should point out here that this is still autosomal, which means that it is not carried on a sex chromosome because, again, we see males and females affected at more or less an equal rate. Right, we have 4 affected males and 5 affected females. That's pretty close to 50. Okay, any questions on Mendel in general? Segregation, independent assortments, monohybrid, dihybrid. All right, are you ready for it to get more complex? Come on, for upward challenges. Yes? You guys can do tough things. I know you can. All right. Okay, so we are going to move into chapter 4, which is taking these ideas of Mendelian inheritance, and first we're just going to adapt them to some situations that appear non-Mendelian but actually are Mendelian, and then we're going to throw them out entirely, and I'm going to show some examples of non-Mendelian inheritance. Okay. Does anybody know off the top of their head any sorts of inheritance that is non-Mendelian in nature?
I like the torty cats.
That's Mendelian, yeah. But we will talk about them. Yeah,
Isn't that like sex determination?
also Mendelian.
What about those like certain animals that like, or sea animals that can choose their sex? Is that Mendelian?
They don't quite choose, but yes, I know what you're saying. That's still Mendelian because they're still getting chromosomes.
What about chameleons?
Still Mendelian. All right, let me ask this question. How many genomes do we have in each of our cells? How many genomes do we have? How many genomes do we have in the human body? No. We have 2 complete genomes in each of our cells as humans. One of them is what we've been talking about. It's billions of base pairs in length, so and it gives us almost all of our traits. But how many people here have mitochondria? Yeah, only 2. Well, the rest of you probably should not be alive. Yeah, so our mitochondria, which all of us have, I don't know if that's news to you, but we all have a mitochondrial genome. It is its own genome. It's about 16,000 base pair in length. It is separate, so it's not in the nucleus of our cell. It's actually in the mitochondria. We may think that it doesn't do a lot for us, but it's the entire way that we produce energy. Right? We use that genome. There are 13 protein coding genes in that genome. It is important to us. If you have mitochondrial dysfunction, then you have issues, just like if you had an error in your traditional chromosomal set. Where does your mitochondrial genome come from? You get it from your mom, right? You don't get it from your dad's. Well, very few people get their mitochondria from their dad. A few people do. We'll talk about it. It has a funny name. It's interesting. But yeah, so you have 1 copy of your mitochondrial genome. It is just from mom. In any way is a unilaterally inherited genome Mendelian. Yeah, there's no crossing over, there's no segregation, there's no nothing. You get it from your mom. She takes her mitochondria and she puts them in the developing zygote, because otherwise that developing zygote would have no energy to develop. And now you have your mom's genome. So if you sequence your mitochondrial genome, it is identical to your mom's, unless you've mutated something. So that would be an example of non-Mendelian inheritance. How many genomes do plants have? 3. They have a chloroplast genome, a traditional genome, and a genome. Okay. So these are sort of the categories that we'll talk about, right? So we'll talk now about sex chromosomes and dosage compensation, which is where we'll talk about our 20s. Then probably on Monday we'll get into types of dominance, and so I assume that you probably are familiar with some of these, like codominance would be our AB blood types, incomplete dominance would be a pink flower that results from the mating of a white flower and a red flower. Penetrance and expressivity we'll talk about, polydactyly again. Lethal alleles are our Manx kitties. We use cats a lot as examples here. And then we'll talk a little bit about gene interactions and then sex-influenced traits. This last piece, our environmental effects, are going to be non-Mendelian. Right? So this is how identical twins aren't really entirely identical because they have different environmental influences that change how their genes are expressed. All right. So in order for something to be Mendelian, it has to follow these 2 laws, which we've already talked about. Does anybody want to define the law of segregation? Somebody describe to me what the law of segregation is, or does, or says. All right.
Is it that alleles randomly unite at fertilization?
Kind of. One step before that. Yes. So 2 alleles, which would be found on chromosomes, when we're going through meiosis, are going to split, and each gamete is going to get 1 copy of each gene, which again are found on chromosomes. So basically, each gamete that you make has 1 copy of each chromosome present. Okay. What about independent assortment?
Yes, they have to divide independently from each other unless they're on the same chromosome.
Yes. So with independent assortment, what your mind should sort of go back to is those chromosomes that are aligning on the metaphase plate and color code them in your mind, like mom set, dad set, and then realize that we can get any sort of combination of mom's chromosomes and dad's chromosomes on either side, so that when we go to the gametes, each chromosome is separated independently of the other chromosomes from that particular parent. You don't get all moms in one and all dads in the other every time. All right, so these are sort of hard and fast rules for the most part. We don't really see a lot of examples of that not happening. But when we're looking at a particular trait in a particular organism, what are we actually looking at? Are we looking at a genotype or are we looking at a phenotype?
Traits, phenotypes.
Right, so we're usually looking at a phenotype. And in some cases, when we have simple Mendelian inheritance, it's very easy to look at those phenotypes, right? That's where Mendel got really lucky. He picked phenotypes that were one or the other, and that all looked very Mendelian. These are traits or phenotypes that are determined by one gene that has two alleles. Okay, so for everything we've talked about right now, you're either big A or little a. Is there going to be any case where you have big A, and littler a? Like 3 alleles for a particular trait?
Sure.
Yeah, there are lots of times where we have more than one allele for a particular gene. We simply boil it down to one or the other because it's easier for us to comprehend. And we've always looked at simple dominant, simple recessive. So if you have that dominant allele, you always show that trait. Again, not always the case. So when we're talking about extensions of Mendel, again, when we look at the chromosomal level, these are still inherited in Mendelian fashion, so they still obey those two laws. The problem is that they become phenotypically more complex. Right? We sort of switch around what's going on with simple or dominant and recessive or with the number of alleles, and it becomes problematic for our brains. All right, so 3 sort of additions to our understanding of alleles, because you'll see these terms in a lot of questions that you're asked to answer. Wild type alleles, pretty sure that's written in your worksheet somewhere this week or next. A wild type allele is simply sort of the most expected allele, or an allele that's really common in a population. And the important thing about wild type alleles is that they are going to give us the proteins that function normally. Sort of the opposite of a wild type allele would be a mutant allele. Mutant alleles are usually very rare. One, because if we're looking at a mutant allele, it probably has sort of just developed in a population. Or it's considered a mutant allele because it's found in low frequency and causes a change in protein production. Which means that it's non-functional. Most of the time, these mutant alleles are recessive. So most of our major diseases or disorders that we think about in a hereditary fashion, again, are recessive. And those dominant alleles, or those wild type alleles, are going to be more common and be expressed more often. All right, so we have talked a lot about these ideas in an individual. We're going to keep talking about these ideas in an individual, but we're going to start adding in sort of a population piece to this. So I probably will try not to use this term a ton, but a genetic polymorphism is going to be a population that has 2 or more alleles that occur in what we call appreciable frequency. So that would be, if we're looking at an entire population and we're sorting alleles, we would have 2 alleles that occur pretty often, 2 or more. Those would be That'd be a population that is polymorphic.
So what's albinism? Example of?
A mutant recessive. Yeah, so if you have a big group of these penguins and you're a predator, who do you see best? Yeah, this little guy. Yeah, so he's likely to just get.
He's in a zoo though, he's fine.
Also, albinism usually comes with other, it's like linked to other things that are detrimental in themselves as well. Yeah. Okay. So we have, I think, briefly talked about our sex chromosomes. I'm going to give us a little bit more information about them, and then I'm going to start talking about sex-linked traits. So there are a number of ways to determine sex, right? And this is, again, biological sex. This is sort of at the fetal and developmental level. We talked about these, right? So this is just sort of a reminder that not every species uses an XY system, and that not every female in every system is going to be our homogametic sex, or not homogametic, what's the word? Whatever. There are not, in humans, females have two X's, right? So we have two of the same chromosome. Here, a female is going to have two different sex chromosomes. Heterochromatic, that's all right. It's something to do with coloration. All right, what else did I need to say here? Oh, okay. So if we look at this figure of the X chromosome and the Y chromosome, which one is bigger?
X.
Yeah, the X chromosome, the Y chromosome is much, much smaller, right? It carries fewer genes. When we are looking at meiosis and mitosis and we're talking about our tetrads, I guess when we're looking at meiosis and we're talking about our tetrads aligning, how does chromosome 1 know that it needs to align with chromosome 1? Okay. They are similar size. What else? All right, so after replication of our chromosomes, we have 46 sister chromatid pairs kind of wandering around the cell and like bumping into each other and trying to find their other sister chromatid pairs so they can form their tetrad on the metaphase plate. So when they bump into, chromosome 1 bumps into chromosome 2, how do they not stay there? How do they know to sort of move on to find the next chromosome until they're paired? Okay, so there are a couple of things. Our sister chromatid pairs of the same chromosome are going to have similar sequence, right? So they can identify sort of the genetic piece, like, oh, we're similar sequence, we should stay close to each other. Another major factor is going to be centromere location. So we have chromosomes that have centromeres at different places. Okay, so if we have a big, long chromosome that has a centromere in the center, and it bumps into a nice, long chromosome that has a centromere close to the top. If the centromeres don't align, they're not going to stay next to each other. Okay, so centromeres are going to be really important. When we are talking about the X and Y chromosome, are they homologous? No. So how does an X chromosome and a Y chromosome know that they need to pair up on the metaphase plate during meiosis? No. Fortunately, they're the last ones left. They're the reject chromosomes. You guys just read the box. Okay, so at the ends of our X and Y chromosome, they have what are called pseudoautosomal regions. These pseudoautosomal regions are like identical in the X and Y chromosome in terms of sequence and structure. and they actually use those pieces to identify each other. Right? So we have the pseudoautosomal region of the Y chromosome actually matches the pseudoautosomal region of the X chromosome, and so they can properly identify the other chromosome during meiosis and line up. Right? This is not an issue in females because females have X chromosomes, so one X chromosome can identify an X chromosome. But in a male, in meiosis, you don't want both sets of sex chromosomes lining up in one cell, right? Because then when they divide, you end up with 4 sex chromosomes in one and no sex chromosomes in another. So there are also non-chromosomally based sex determination systems. Some of them are what we call genic. So in this case, there are no chromosomes that are specifically linked to sex, but rather a set of genes that will tell a developing embryo whether it's going to be male phenotypically or female phenotypically. Okay? And so these are going to be common in plants, fish. Everybody knows Nemo, right? And Nemo's dad. What's wrong with Nemo's dad? Yeah, so Nemo's dad, when Nemo's mom is killed, or we don't know what happened to her, but when she goes away, Nemo's dad should have become female. They're called sequential hermaphrodites, likely as a result of a genic sex determination. You can turn off and on genes and change phenotypes based on the circumstances. So in that case, a male would actually become female, not because the sex chromosomes changed, but because a different set of genes is turned on under that environmental condition. Yes.
So is that considered non-Mendelian, or is that still Mendelian?
It would still be Mendelian because the genes are inherited in a Mendelian fashion. We just change what is actually turned off or on. Right. Okay, and then we have environmental factors, which are usually temperature related. So where you are in the environment, if you're close to the outside of a stack of eggs, you're going to be cooler. If you're closer to the center, you're going to be warmer. And those will then determine whether you become male or female. And I talked about this, so I'm going to skip that ahead. Okay, so what is it about the X and Y chromosomes in females that determine whether an embryo becomes male or female phenotypically? The answer is a particular gene that is located on the Y chromosome. So on the Y chromosome, we have a small gene called the SRY gene. And the presence of that SRY gene is what determines maleness in a developing fetus. So it's not the whole chromosome, it's not like a whole bunch of genes, it is the presence of this particular gene. This particular gene is going to start coding for testes development. Testes then make testosterone. Testosterone is going to change the rest of the phenotype of the fetus. So, these are just my fun facts. When does sex differentiation happen in humans during fetal development? No, like in a developing embryo. When does it become?
When would you be able to tell? What were yours?
It's like 6-ish weeks. So it's like, yeah, right about halfway through the first trimester. That's 4 weeks after fertilization. So for the first 4 weeks of fetal development, everyone's a female. And then testes and testosterone produce males. All right, when we have errors in meiosis, we can end up with different combinations of sex chromosomes. Unlike autosomes, we can actually have too many or too few sex chromosomes, and it's usually not a huge deal. There are going to be some phenotypic impacts, but overall in terms of like development and intelligence and things like that, tends to be minimal compared to external autosomes. And so these are examples. These would be our sort of standard, what we would expect, XX or XY. In some cases, you'll get 2 X's and a Y, 2 X's and 2 Y's, 3 X's and a Y, right? These are some pretty serious meiotic errors that would cause these types of phenotypes. This is called Klinefelters. The presence of the Y at all results in a male phenotype. Here, these two, we have only X's. Apparently you can get up to 5 X chromosomes in a fetus. These, regardless of how many X's you have, whether it's 1 or 5, phenotypically female, right? And then here, 2 Y's, phenotypically male. So anytime you have a Y chromosome, whether it's 1, whether you have 14 X chromosomes that you think would make that effect minimal, still going to be phenotypically male because of the presence of this SRY gene during fetal development.
How do you get an XO?
So you would have a sperm that contained no sex chromosomes, or an egg that contained no sex chromosomes, and then the other parent just had 1. All right. Set that. Okay. So I showed pedigrees that looked at just autosomal recessive and autosomal dominant traits or diseases or disorders. There are also sex-linked diseases or disorders. So everybody's favorite to talk about is red-green color blindness. It is a sex-linked trait. Male pattern baldness, sex-linked trait, right? Those are linked to the X chromosome. When we're talking about genes that are present on the sex chromosomes, which one is more likely to have a mutation that's going to impact people? X. That's the question, Y or X? Why would the Y chromosome be more problematic? All right, so are mutations more likely to accumulate in this chromosome or in this chromosome? In the X chromosome, because it's bigger, there are more genes to potentially be affected, right? The male chromosome has like 20 genes. Some people aren't even sure why it still exists, because it doesn't actually carry genes for many things except for phenotypic development during fetal stages. The X chromosome, we use a lot. It has a lot of genes on it. There's like 200 genes on this chromosome, so we use it for a lot of things. If there's a mutation in an Y chromosome gene, it's going to affect more people. Right? How many people in this room have Y chromosomes? Not to point you out, but a few. How many of us have X chromosomes? All of us. Okay. So X-linked conditions are more common. Start standing still. I was in A100 and I was teaching Bio 100 and I fell off the first step. I've never heard a class pay so much attention to what I was doing. And then yesterday I fell. So embarrassing. I fell while I was running and I almost rolled into the street. And I broke my shoe and my key and my headphones. And I couldn't write anything last night because my hand hurt, and I couldn't wear pants today because my knees are skinned. And now I'm falling for you guys. This is why you don't run. I get really angry when I don't run, so I try not to get angry. Okay, so I'm just gonna stand still. So, what was I saying? X chromosome, more common to have X-linked conditions. Males are more likely to be affected by these traits than females. Why would that be?
Because they don't have the extra copy that can prevent the mutation.
Yes. So if we think about a female has 2 copies of an X chromosome. So if one of those copies has an X-linked recessive trait, right, say color blindness, for example, and the other one has a normal allele, which is the dominant allele, she is going to show the dominant phenotype. Everybody with me? Okay. If a male inherits an X chromosome with that mutation, on the Y chromosome, there is no gene for color seeing sight, right? So they only ever express the mutant gene, right? So males are more likely to be red-green colorblind, and other conditions that are carried on the X chromosome are more likely to be seen in males. There are some genes on the Y chromosome, but when we talk about inheritance of the Y chromosome, it's only from father to son, and so we have a smaller portion of the population that's likely to be affected by those. And there's also fewer genes, so just by chance, we're likely to get fewer mutations and have fewer issues. Okay, so I'm going to quickly walk through one of the first experiments that showed X-linked conditions, and it was done in fruit flies. Right? In this case, we are looking at eye color. Okay, so most fruit flies, if you see fruit flies in your house and you look at them, like I do every time I see a fruit fly, I look at its eyes before I squish it. Usually they're red-eyed. That is going to be our wild type. Okay, red-eyed. So next time you see fruit fly, like think, I'm not just weird. Like take a look at it. I'm also weird, but it is interesting. So. Actually, for this experiment, this was done by Thomas Morgan, who is going to be one of the sort of leaders in fruit fly experimentation. And he was trying desperately to induce mutations in his fruit flies because he wanted to try these sort of interesting crosses. And so he did all sorts of crazy things. He took fruit flies and he reared them in the dark, hoping that the dark would cause mutations. And then, what else did he do? He exposed them to radium or X-rays, and again, trying to find phenotypic mutations. And then eventually one day, after many, many years of attempting to get mutations showing up in his fruit flies, he found a white-eyed male, which is a mutation. And so he took it home with him to make sure that it didn't die or mate or do anything weird, and kept it with him until he was ready to run experiments on it. But he ended up coming up with really cool results from that little pet fruit fly male that he had. So, it's not very long.
I didn't think so, so I was like, take it home.
It's not very long, yeah. All right, so in this experiment, he took a red-eyed female, which would be our wild type, and crossed it with our mutant male. And the results looked like this. Okay, so between that cross, he got all F1s that had red eyes. In his F2 generation, he got this outcome. All right, so if we look at this just kind of on the surface, does this look like what we've talked about before? Yeah, this looks pretty much exactly like what we've talked about before. Okay. He kept going with it, and eventually he got a white-eyed female, so if you keep mating after the F2, you'll end up eventually pulling up some white-eyed females. When he crossed a white-eyed female with a red-eyed male, his F1 suddenly looked different, right? Both of these individuals do not have the same phenotype, right, which is what we would expect in an F1, but instead, every female is red-eyed and every male is white-eyed. That's immediately a departure from what we've seen in our other F1 generations. In the F2 then, we have 25% red-eyed females, 25% red-eyed males, 25% white-eyed females, 25% white-eyed males. Again, if we just look phenotypically at the divide and we ignore the sex factor, we have 50-50. Again, not what we would expect from a standard monohybrid Mendelian cross. Okay. And so the difference between traditional or simple Mendelian inheritance and this is that the eye color is actually linked on the Y chromosome, and so we have to add the sex factor in. And so on, let's see, yeah, maybe on Friday or Monday I'll talk more about this piece. So we can get through the rest of our material. All right, let me know if you have questions on the worksheet. I'll be around some of tomorrow, and then definitely in class on Friday too.
I will not be the one.
But it was like, it's gonna be a sort of like. So it's this thing only. I don't What character is this? Would be like that.
Yeah, I'll see you later. I turned it in on Monday.
So what I was looking for here was what we talked about with Mendel, the fact that.
Will you help me? That is something with my education wrong
So
right
like, I
now.
don't know what.
And like the only way that I can do
Mendel
it
is actually. It's too late this semester. Really? Maybe only in humans, but like typically distinct, like you can tell something when they're alive. But when we look at, it's actually.
We're going.
Oh, but there's no money involved.
I obviously like one, and then I'm like, I probably shouldn't make that. Too many. Sorry. I can't. And we only.
Did you say you're gonna take it?
I have a meeting, but I have to go work out.
Yeah, we're going to a meeting from 12 to 1. We're cutting puppies at 11:30.
Alright,
The puppies on campus there.
I just wanted to do this the whole time. We're gonna hit the video also, just give me some eye, just like.
Puppies at 11:30. And then